My name is Fiona Quenano. I'm part of Group 2, and this is a gut microbiome experiment for Bio 200. A little bit of introduction. A microbiome is a collection of microorganisms that inhabit an environment, creating a little mini ecosystem. Our body is made up of many different microbiomes, each unique in all parts of the body. Our gut microbiome is used to break down and absorb nutrients from the food we eat. Without it, we wouldn't have the nutrients needed to function. One physiological condition related to the gut microbiome is obesity. Obesity is related to metabolic disorders where the bacteria can't break down the food and absorb nutrients. Energy imbalance, regulation of energy storage are all related to the gut. Hormonal imbalance is a characteristic of our gut microbiome as it produces estrogen. If the gut microbiome is unhealthy, it may lead to a disruption in estrogen homeostasis and development of chronic diseases such as cardiovascular disease or even cancer. In this experiment, we are testing to see the similarities and differences in the gut microbiome of a herbivore and a carnivore. Our hypothesis is the bacteria will vary between a herbivore and a carnivore due to the difference in their diet. The animals we are comparing are a 2-year-old iguana, which is our herbivore, and then a 2-year-old snake, which is our carnivore. Right now for our materials and methods, step 1 was the isolation of DNA with the QIAGEN kit. Step 2 was the DNA concentration with the Zemo kit. The DNA will then be 10 times more concentrated. The Applicon PCR gene that was amplified was the 16S RNA gene. For our positive control, we used a known concentration of E. coli, and then for our negative control, we used water. Step 3 was the quantification of the DNA using the nanodrop spectrophotometer, which measures the impurities of the DNA, whether it's contaminated or not. DNA absorbs light at 260 nanometers, while protein absorbs light at 280 nanometers. If the ratio is below 1.5, then your sample is contaminated. Step 4 is the gel electrophoresis, which is used to separate the DNA fragments by size. The smaller DNA fragments will move further than the large DNA fragments, moving from a negative to a positive side on the gel. Step 5 is the PCR cleanup, where the DNA binds to the beads, and then we use ethanol to remove all of the impurities. Next is step 6, which is the index PCR, which helps to determine the nucleotide sequence by adding a unique sequence tag to each sample's DNA. Step 7 is a second PCR cleanup where we get rid of all the impurities. Step 8 is the fluorometric DNA quantification, which helps determine the concentration of the amplified sample, which is more accurate than the UV because it's more sensitive in distinguishing between the RNA and the DNA. Then step 9, we determine the molar concentration of the index PCR product. Step 10, we prepare the dilution of the DNA for sequencing, and then step 11 was the actual DNA sequencing. Alright, now for our results. Here I have the UV spectrophotometer readings. The DNA extracted was successful in terms of quantities for sample 555, our 2-year-old snake, and sample 410, our iguana, meaning there was more DNA than protein extracted. For our sample 429, our 12-year-old snake, from the UV spectrophotometer results, had a low DNA concentration, meaning we could not use that sample for further analysis because there was not enough DNA to analyze. Here we have our fluorometric quantification results. The concentration reading for the fluorescent spectrophotometer were much higher than the UV spectrophotometer, which is expected because of the fluorescence is more accurate. The molar concentration shows how much nanomoles of DNA is in our sample. The concentration reading for 410 was low for both spectrophotometers, but it still had enough DNA to analyze and to continue on with the experiment. Next, we have our reading results. There might have been slight contamination in our results because our negative control had a reading of 452, which is a low enough reading compared to our positive control, which had 12,284 readings. Again, this is a low enough reading that our data is still good for analysis. Here we have our gel results. In the positive lane we have E. coli, in the negative lane we have our water, and then in lane 1 we have sample 555, and in lane 2 we have sample 429, lane 3 is sample 410. The gel shows the amplified 16S RNA sequence for sample 555 and 410, both resulting in a good reading. The DNA fragments migrated, revealing a band in a specific sequence. Sample 555 had a distinct band, and sample 410 had a faint but noticeable band. I don't know if you can see it there. And then sample 429 did not have a band, meaning the DNA concentration was too low or there was contamination, which is what you could see in our UV spectrophotometer results, which is why we crossed out for sample 429 from the beginning. Our positive control had a distinct band, which is expected because that was our known concentration of E. coli, and then our negative control had no band, which is a good thing, indicating no contamination in our DNA samples. Here you have our phylum classification. Here you have sample 555, sample 410, our positive control, and then our negative control. Protobacteria, bacteroidetes, and firmicutes were found in both samples. As you can see in sample 410, there was more bacteria found than in sample 555. Sample 410 is our herbivore, our iguana. In our positive control, there was only protobacteria found in the sample. Our negative control had a low reading compared to our positive control. Here I have our class classification. Again, sample 555, sample 410, our positive control, and then our negative control. Sample 555 has more gamma protobacteria than Clostridia, and our sample 410 is kind of the opposite. It has more Clostridia bacteria than it does gamma protobacteria, and then our positive control was mainly made up of gamma protobacteria, and for our negative control it was gamma protobacteria and Clostridia bacteria were kind of split in half, so I thought that was kind of interesting. Our negative control had quite a few bacteria, but not a lot compared to our positive control reading. Here I made a similarities and difference chart for the phylum class. As you can see, protobacteria is found in all samples, firmicute were found in all except the positive, and bacteroidetes were found in all but, again, the positive. And then sample 410 had quite a few bacteria that sample 555 did not. Many protobacteria live symbiotically with plants in their roots, and they can be found almost anywhere. They include a wide variety of pathogens such as Salmonella. Others are free-living, which may include bacteria responsible for nitrogen fixation. Nitrogen fixation is symbiotic with plants. Gamma protobacteria, which was found in our class classification, can be found in Salmonella. A high-fat diet promotes an increase of firmicutes and bacteroidetes, as shown in our phylum class. A high-fat diet can be related to obesity, which is what I discussed earlier in our intro, which can decrease your metabolism. You should have a healthy diet to maintain homeostasis and a balance of firmicute and bacteroidetes. So, in conclusion, our results kind of make sense. I mean, if protobacteria can be found in a plant, so if our herbivore ate a plant, you'll find protobacteria, which kind of explains our sample 410, which is our carnivore. If, say, the carnivore ate a meat that ate a plant, then therefore you'll find protobacteria in our carnivorous sample.